So we have only about 10 minutes delay. I'll try to do my part by being fast myself. But just to give a recap, what we are going to do now is we're going to look a little bit based on data type division, give presentation for each group, go from sequence to structure to images, and of course all crossover because we are working horizontally. And I will start this off by giving you some update of what we are doing. These presentations are meant to encourage collaborations, both from people listening now or people clicking on the link later. So please always feel free to contact us, and we are happy to work on different things. I also will touch on the virus part in the second half of my talk, and not so much on the case numbers. It would be difficult to follow Michael on this one, but very much on the sequence-based side, also related to the variants. So in my group called Protein Sequence Analysis, we have a common scheme. So we look at sequences and we try to make sense, add value by understanding the function. And this function often is manifested to go from the sequence, from the nucleotide to the protein to the structure. And we are not doing detailed structure modeling though. So this is done by other groups. At the same time, at the sequence level, you also can gain insights by comparing the sequences with each other. And of course, that is also very important for several aspects. We do work on quite of a range of different topics. And the underlying same methodology to look at sequences, changes in sequences, and the possible consequences on functional changes is what we're doing. I'll start here on the corner with our work, where we are also working with SIFB and previously with also SUBI and Yoga on some of the enzymatic ways how you make small compounds. This could be in the food space, but also could be at the level of drugs. And we have a long running program in Singapore, the Pharma Innovation Program. And my staff, Joanne, is our soldier that during the pandemic kept producing important progress in our ability to predict effects of mutations on the enzyme function. We have a joint project, including inside BI with HOW, with ISIS, with the new name, IHBC and SIFB. And it's a combined experimental and computational effort. And we are trying a lot of different things, including using new AI methods like CNNs to predict binding free energy in the binding pocket here. This is the galactose oxidase. You see it has two domains, the beta propeller here, binding pocket is here. So if you focus on, for example, if experimental data from about 2,600 mutant to substrate pairs, experimental measures, what it does, we can learn from it how well, for example, one chemical substrate binds when there is a mutation or not in the binding pocket. But also very importantly, when you want to look at the performance of an enzyme, mutations away from the binding pocket may affect. And then there are different reasons how they can affect. This could be effects on stability. This could be effects on aggregation so that the whole protein clumps together. There could be allosteric effects using the allosigma tool, for example, from Igor. And there is also non-structural features that we can utilize. For example, the sequence conservation. Evolution gives us a signal to know the importance of some positions and also the criticality if you would mutate them. So just to summarize here a few of different things. Joanne has been trying different methods, Foldix, one of them, Yasara, at some point later also switching to Rosetta. And I think it's very important to try different things and understand if you feed different features into your machine learning models or neural networks, how this affects the outcome and the fitting that you do. And she has found that stability can be predicted in several of the cases, including increase in thermostability, the top 3 binders. In terms of the allostery, among those that were found experimentally to have a long-range effect, 7 out of 14 were also predicted to do so using Igor's allosigma tool. And in terms of aggregation, I would say this is a bit more mixed. These are two tools that I also co-developed previously, and especially Vaults. And when you combine the effect of, let's say, the tendency to aggregate after mutation is done with the change in stability, you can get some leverage on solubility. And although there is not the large-scale correlation, I would say, there are abilities to find much more soluble proteins, and this is what you want in the end. So what this project does is try to predict which mutants are likely going to work for which kind of substrate, and work in the sense that the protein needs to be soluble and have a high yield in the end. And combining this data is the last step that we are going to do to try to make a more integrative model. And this is, again, a highly collaborative work. And I think Hao will also talk about it with the docking scores and several other features. So the next point I wanted to mention very briefly is that also at the what looks like the verse, like shampoo or the new impossible burger, what how does this relate to the kind of work that we are doing? Very simply, when you have a sequence and you want to know if another sequence is similar, and the feature depends on the 3D surface structure, like for an allergen, it's also antibodies that bind. Just like when the flu virus detects with IgG antibodies, in allergens, it's mostly IgE antibodies that recognize. So what we've built a while ago in the first version is a tool that can predict similarity, including sequence and 3D structure information of the epitopes. And especially over the last year, both Vachi and Min also from Chandra's division, have worked on improving this tool. So we are now on the verge of AlaCAD Pro 2.0, where they include more information that has become even better. This tool is important because it does provide ability for companies, P&G co-developed this with BI, and to assess if a protein in a product is likely to be allergenic or not. And the tool is also used by EFSA, by the Singapore Food Agency, and this provides some additional support to evaluate also the safety of novel foods. The third item I want to mention is again collaborative. So you can see already we are at BI often working across groups and very horizontally. This is a particular effort, of course, also across different institutes with GIS, but also with Chandra and Asha, combined with my staff Dimitar and Ken. When in Singapore, there is a national precision medicine program that has already sequenced 10,000 healthy people, and now is on the verge to 100,000 and then later 1 million. And we also want to know, just like I said in the beginning, if there is a mutation, what does it mean relative to the binding site of some drugs, for example? This can happen in molecules that are drug targets or that are metabolizing drugs, so that they will change the efficacy of the drug. And in the age of precision medicine, having the genome alone is not enough. You need to be able to tell what do the changes mean. And using a combined workflow of different data sets from PDB, ModBase, HHspread, and AlphaFold, including BLAST of sequences from a human reference proteome, and the team around Asha as well created this annotation that we know now if a position in a human protein either has likely no structure or no predicted structure, if it has some structure known or predicted, and if it has structure, if that position is near a drug or not near a drug. And this annotation does take quite a long time to calculate. This is made available as the SNP Drug3D database. And the statistics are summarized here also by a predicted phenotype of the amino acid substitution, for example. I will not go into great detail here, but this is a paper that is just being submitted now. But I think it's interesting to see that for the first time, for the whole human reference proteome, for every position, in principle, we have the knowledge if there is structure, no structure, as well as near drug and no drug. And AlphaFold also contributes, of course, here, but it doesn't do everything yet. And it's we are bringing the field forward by adding the notion of the vicinity of drugs. And then Dimitar has built the database interface, as well as the backbone of the database, where you can browse and look for this. For example, CYP2C19 as an example here. So you can access CYP2C19 is cytochrome P450s that are very important for drug metabolism. And here you see then a more detailed view, including the structure. It sounds relatively straightforward that, okay, we know there are SNPs, you can map them through to 3D structures, but until recently, until this time, there was no tool available that could tell you if that SNP is near a known drug or another ligand. And this is the first systematic study where this has become available. And Incomes also Ken, who did the PhD with me, and he did a very nice tool, taking the cytochrome P450s and predicting if the SNP in this protein family not only cause a disease or neutral from a very carefully curated benchmark set, but also if it has pharmacogenomics effects. So if the drug binding affects the efficacy or the turnover rate, for example, of a drug. And last but not least, in this whole project, together with Chris, who is in Shenzhen, but also has a team in EDDC, together with Judith, they experimentally verified some of the predictions that we found. We pick here exactly CYP2C19, one of the enzymes that metabolize many of the drugs that humans take. And this is clopidogrel, for example, a blood thinner. And we could see and verify the effect experimentally of the single nucleotide polymorphisms on the drug binding through these experiments. And now this brings us to the part that we have worked as a larger team the most. You've already seen our different people and across the institute helping in different projects. This, of course, also happened here. I list here below the members from my team, as well as Shruti, who is also across our different divisions. When Michael, just before, was talking about the case numbers in different countries, all these different outbreaks, they are also characterized by some changes in the virus. Many of these changes are simply license plate changes for us to know that the virus has a neutral marker that we can describe it better. But in some cases, it also changed the properties of the virus. And we have been, since the very first appearance of the virus and the sharing of the sequences by China CDC with GISAID on 10th of January, on the forefront in helping to look at these sequences, make sure they're looking good, scientifically sound. We do this through quality checks, curating them, and then using the GISAID platform, that is the unique platform that protects the user's rights to their data, but allows sharing in real time. And we are helping bringing this through. It also started with our curation team here, but very quickly became a global effort again, where there are teams in South America, in Europe, in Africa, in Asia, working together to have all the time someone available to look at sequences, bring them through. And this has resulted, in fact, in an unprecedented wealth of sequence data, more than 9 million. We will soon reach 10 million. And this amount of virus sequences actually is bigger than any other database before. And it's really a clear sign that the GISAID sharing mechanism, where you need to register yourself, but everyone can access it freely to see the data and share the data. And you need to acknowledge if you're going to use the data. So you cannot scoop someone else with their data. This really incentivizes rapid sharing. And this rapid sharing has enabled that we could see in real time all these variants emerge. I want to show a few efforts here. For example, all the vaccines and diagnostic kits already were started with the first sequences, including in Singapore. But also it's important to check if any of the new variants, the changes that are caused, would change the way the diagnostic kits work and perform. And this is done on a regular way. One version of this was written by Winston, and Joseph in my team. He's doing not just this regularly and writing reports twice weekly, about if there's any change and helping to monitor this. But he's also looking at creating maps to see submission patterns, submission delays, to know where the sequencing capacity possibly could be increased. Another important effort is that we can do with the sequences. We can look at how are they related to each other. In the very beginning, in the first 3 clusters in Singapore, one of them was with a traditional Chinese medicine, tourists that came from Wuhan that were there. It was very interesting to see that we could overlay the epidemiological and genetic networks, and they were matching quite well. And of course, this was done manually. Sandy in my team, she was also at the forefront at the National Public Health side. And since she joined our team, she continues with the effort now on the computational side. And this includes for us to look and compare at the different genomes at the ASEAN and also all global level. At the ASEAN level, we can see in blue our Singapore sequences, how our cases are likely to come from. This also includes for us to know if we have local transmission, if we have imports, and if any measures that we apply work, if we can control the outbreak. At the same time, because the data set has become so big, a simple blast search would take too long. So we are developing new tools to do this much faster. Now the backbone of a lot of our work and support for GISAID is something that started in 2009 with the flu server, which I wrote for H1N1 pandemic. But for this coronavirus, the same code was fully adapted by Raphael in order to make it work for coronavirus. And in a similar pattern, what we have here, it's a database where we have manual curation of literature for phenotypic effects of mutations. We have statistics on occurrence of mutations. We have 3D structure information. If it's near antibody binding sites or receptor binding site, this was contributed by Asha. And the combined information allows to give you in 1-2 seconds view the importance of a mutation in the virus. And having this system around actually did help us. In early April 2021, we were looking at a new subclade in a tree that came from cases that traveled from India to Singapore. And when we looked at it in the structure, we saw there were receptor binding changes. There were antibody binding site changes. And so we also connected with our colleagues in India, for example, the INSACORP consortium. And they also already started looking at this new variant. This variant then later became Delta. And we were discussing that this is something to look out for at this time. And then later you see the rise of the curve. This is where it went. And very similar in the case of Omicron in South Africa, the team of Tulio has always been very well aware, together with the clinicians in the network. They already had found another variant of concern previously. And when Omicron came, he sent us an email to ask for some help to do comparisons. It looked so different with so many mutations. I thought it was a sequencing mistake, but they confirmed it was sequenced by 3 different methods, and there was no way also of being able to hold back this variant anywhere in the world. Very soon, when they reported it, it was already everywhere. And because of the ability to quickly act, the team, including Winston, Yanni, Raphael here, they have built trackers to allow to very quickly visualize where the latest cases are found, etc. And very soon this gets overloaded, and you will see it's essentially everywhere, and it follows this curve. So the most interesting part we're working on currently is this integration of the different data feeds that we have, the phenotypic role of mutations that we have from COV server, the rise in global distribution because of GISAID's real-time surveillance. And this is summarized in graphs and new ranking algorithms. We have this tool called emerging variants. And Raphael and Yanni are working on this interface. Joseph is always also preparing reports. And we are reporting to agencies like CEPI. We are discussing with WHO. And then there are from these findings of what is a new trend of a variant, people are doing experimental tests to test if the vaccines still work, etc. So it's pretty much an important system that will go on. I want to highlight again that the virus naturally always changes, and most of the mutations of a virus are just like a license plate. So we know where it came from, which also can help a bit understanding transmission and the control of the virus. But in some cases, it's also phenotypic mutations. Then we have different properties like in between Delta and Omicron. And these are then some changes that, although the virus looks the same, except for a little change, maybe the engine inside is a bit faster. And our tools are contributing globally to hopefully give a realistic assessment of when we should look at the virus in a different way. But all of it is, if it's a prediction, has to be verified in more detail. And to conclude, I'm very grateful to our whole team, including the global GISAID team that is working with us, but especially with the team Singapore that has been really working hard through 2 years. And I would call them frontline workers. We are all part of it in a way. And I also want to acknowledge and appreciate that BI is a great place to work. And it's also the go-to place where we can get real value out of biomedical data. So that's the end of my talk.